No, but with your.
All right, so here you go. Ready? This is a tough one. That one, right? All right, good. But all right, so.
Never really learned about that.
This is really the question I want you to know. We emphasize. I thought I deleted that first slide, but obviously I didn't.
Can I close the blind?
Sure, absolutely. Thank you. Yeah, is it glaring?
Which one is it?
I don't pull one if it go, turn and pull the other. On one side, one goes up, one goes down.
Okay.
There you go. All right. Some of the things that Mendel did well as a result, why his findings were so remarkable is one that we talked about is he really had no knowledge of chromosomes and know anything about meiosis. So, the whole idea of segregation, all these things he found out were pretty incredible at the time. But the experiments that he did were they were controlled, and he did things like snipping off the male parts of the flower to make sure that there wasn't any cross-pollination when he was doing self-fertilization, things like that. He did huge sample size. Remember the numbers we introduced earlier, thousands and thousands of samples and repetitions of the same experiment. And again, it was verifiable and repeatable. People since then and still this day are doing the same kind of things with different organisms, coming up with the same type of results. So those are really big deals. We, at the beginning of the year, talked about this. Remember some of the things that made good experiments? So again, just to kind of carry that theme throughout, I want to make sure we reemphasize it when we can. We saw it here. We'll see it when we get to talk about Darwin and evolution and things, similar things. And it basically, this really is a trend that you see in all good scientific theories, is they have to have this or they would never have gotten to where they got. There's some other things, too. It has to have a testable hypothesis. Remember that. If you throw out some bizarre hypothesis that can't be tested for experiments, it's no good. So obviously his was in such a lot of good experimentation going on there. All right, so when Mendel got done, he came up with two things, laws he called them of genetics, okay? Who knows what they are? Who knows one? Yes, ma'am?
Law of segregation.
Law of segregation. Another one?
Independent assortment.
Okay, very good. Segregation, independent assortment. Super. Okay, so what does that law of segregation mean, Jake Kelly? You want to pass?
I'll pass it to you.
All right. I figured you knew that's where I was headed.
All right.
You can do this.
Does it mean that? Okay. The chromosomes go to like the 3 to 1.
Nope. Who's going to bail them out? Come on. This is like the easiest one of all, guys. What does segregate means? Separate. So, Sasha, tell me what this means in term.
Is it like they?
Wow, you guys, this is really
Chromosomes separate in some way.
yeah. Chromosomes separate during gamete formation, basically meiosis. Remember, so when you as the chromosomes, homologous pairs separate, only one of each pair goes into the gamete. That's called law of segregation, okay? So only one allele from each of those pairs is passed on, and that means they separate or segregate. This is probably, again, one of his greatest accomplishments, because, again, he didn't know that. He didn't understand. Meiosis wasn't even the concept that was talked about at that point. But he basically figured it had to happen, otherwise the results he got would not have happened the way they did. Okay? All right. The next law, independent assortment, a little more complicated, but who wants to give this a shot? Alex, give her a shot.
Let me think about it for a second.
All right. Stephen?
traits are chosen separately from one another?
Yeah, yeah. One trait, hair color, may be different from like height. They're independent most times. Now, as we know, there are exceptions to that. It's the linkage situation. But as far as Mendel was concerned with the traits that he dealt with, that's exactly right. It means one trait doesn't affect the other. And the reason is that they're on different locations, and because of, if they are in the same chromosome, crossing over can occur to shuffle them up. Okay? And in fact, in Mendel's case, one of the traits was on the same chromosome, but they're so far apart that crossing over made it seem like they were on two different chromosomes. Yes?
Is the, I don't know what word to use here, but is the opposite epistasis?
I guess I can see how the reason, but really this is more an explanation of the phenomenon that in all cases you have this occurring. I guess not all cases, but the exceptions to this rule would be epistasis and linked genes. Yeah, so this is a normal scenario, and this should make sense
So that's
again.
like the dominant emphasis of narcissism.
Well, not just down. This occurs in all cases, but then once this occurs, then depending on the way the genes are expressed, once they've been shuffled, then that shuffled gene might over-dominate. No, that would be epistasis.
Okay.
So this is a physical happening of all chromosomes, with the exception again of sex-linked chromosomes or X-linked chromosomes, which are linked genes, which they don't assort independently and therefore appear as if they work like a monohybrid cross. So really, to see this happening, you have to have a dihybrid cross, because the whole idea of this is, for example, and we'll get to this later, if you have like a big A, big B, little b, the idea you have the big A can go with the little one, the whole idea of the foil and the distribution of all the different possibilities of gametes, that's due to the independent assortment of those separate alleles. And then because they assort independently and they separate, you got to figure out what are all the possible combinations, and that's why we come up with 4 possibilities for 2 traits in a dihybrid cross. Okay, that's the whole idea of independent assortment. All right, so we get into a dihybrid cross. What is it? What is the difference between a monohybrid and a dihybrid cross here? Okay, Hunter, could you know this one? No? Hey, Will?
Dihybrid cross involves crossing 2 traits.
Yep, monohybrid. That's how you do that one.
Because you have part
Yeah, you need to know that, okay? So, really, remember hybrid means same as what other term? Heterozygous, right. So those terms are linked. Then there's homozygous, and what's the other term that means the same as that? No. Pure. So remember, don't forget the regular vocabulary now, because we actually still have a crossword. It's that crossword we are only testing over 10 of the 15, and remember there's like 38. So don't forget the crossword, and we'll get to that at the end. All that vocabulary is still part of the game, and the stuff we've been working with should just be second nature right now, because really you couldn't have gotten here without knowing the vocabulary. All right. All right. So basically, here's an example. So we got a white, red flowers. So you got this is a monohybrid cross. So you have a white with the red flowers, and they all came out pink. What? What are we thinking about here? Yes, Jake.
Oh, is it a co something, right? Co-inheritance or something?
Close. Incomplete dominance. Yeah. In this case, you're going to see, Jake. Next one. Imagine a previous example. You have the same thing. You have red and white offspring. What's that? Forgot, it's recycling day. Okay, so here you go, Jake.
Oh, okay. Is this co-dominant?
There you go. All right. So again, this is make sure codominance means that the dominant traits of the parents stay and do not change. Incomplete dominance means that there's an intermediate between the two. It's kind of a mixture of the two. Okay. All right, good. All right, so here's another one. Mendel had some pea plants, and then we got pure breeding round seeds with pure breeding wrinkle seeds. So what kind of cross are we looking at here, guys? Dihybrid. Okay, so we have two traits we're talking about, so you should recognize that. So you have two letter combinations. So pick a letter, a big W or a little w, whatever you want to use. And then he got an offspring, okay, and then he took that offspring and crossed two of those, and we did a problem just like this in your Jolly Green Giant and something Sally, remember that? We did that cross, and then you looked at, so what would you expect, how many would you expect to have smooth seeds? All right, so let's go back to that. So the reason is that's a smooth, it's pure breeding wrinkle seeds, and then you fertilize the next generation. But how many offspring would you expect to have smooth seeds in the final generation? So you have dominant trait. And this is second generation, so you're going to have 3/4 of them with the dominant trait. So basically it's a dihybrid cross, 3-to-1 phenotypic ratio. So you should have 75% with that smooth seed. Okay? Actually, this was only a single factor cross, wasn't it? Yeah, I goofed up there, didn't I? Erase. All right, so basically you get two pure breeding traits. Pure breeding sounds like a big T, little t, big T, big T, big T, big T. That cross with little t, little t. You're gonna have all big T, little t's. You cross those two. You're gonna have 3-to-1 of the big T trait, or in this case, the smooth, and then 1/4 of the other. Okay. So 3-to-1 phenotype and 1-to-2-1 phenotype. Okay. So if you did this, you had parents, you had their kids, and you crossed two of the kids. What's that second generation called? Who knows? F2. Okay? So it goes P, F1, then cross two F1s, get F2, and that's usually all father we got. Hand up. I saw a hand up somewhere. Okay. All right. Here's some basic terminology. See if you can fill in the blanks here. Zach, what do you think? Genotype and phenotype. Very good. Okay, that should be pretty straightforward. Just to make sure you got those two the same. All right? Okay, so traits that appear on X and Y chromosome, and what are these guys called? Good? Sex link. Good job. All right, so in sex link, when the mom expresses an X-linked recessive trait, who shows these traits more often in their phenotype? Basically, you have a 50-50 chance on this one, right? Rachel, what do you think? Boys or girls? Say what?
Male, boy.
Exactly right, males, and that's the whole point. And why? Question of that is why? You know this one? Remember? Zach? Hannah?
Because of the XY chromosome.
Right?
And the mother passes it down.
Okay. And you're halfway there. Yes.
He has to receive the female X chromosome because his father's not.
Okay. Jake?
Is it because the Y doesn't carry anything?
Yep.
So
That's it. That's what you're all right. What I want to make sure you do is it makes sure that the Y, again, doesn't have any alleles on it, so it only takes one of the recessive traits to show the trait, whereas in females it takes two. Therefore, the odds are twice as more likely, just by that alone, because again, Y isn't really factored in it, that guys are going to get the trait. All right. That's kind of a key point in the whole section. All right, so Punnett squares is what they use for. This is like a no-brainer, okay, right? Probability. So it's really to show you the possible offsprings from a cross. The top, let's break away from this for a second. In a Punnett square, we'll just